synthesize the entire human genome. No, sorry, we didn't synthesize it, we decoded it. And we actually did it it was in February of, I believe, 2001 or 2002, back in that day. And it was actually, we were able to do that. We were able to decode the human genome one day before a private company did it. And had that private company did it before we did, they would have been able to patent different parts of that human genome. And because we're a public university, that information is now readily available. So the way that relates to a ribosome is that a guy named Harry Noller, who's a professor at UC Santa Cruz, discovered the ribosome. No one really knows it. Ribosomes are really cool because they're involved with that part of the central dogma of biology, which is involved with translation, which is involved with turning that RNA into protein.
Just like going off that, who knows what cosmological thing was discovered at Santa Cruz?
Something about the moon?
The inhabitable planet?
That's what I was going to say.
Or no, it's more important than that.
More important than
Yeah.
other inhabitable
Who's going to control?
planet. Is it ocean water on Mars?
No.
Dude, way more important than space.
It's dark matter. Dark matter.
Oh, really?
Yeah. Dark matter was discovered by Joel Freeman.
What?
Yeah,
Are you
really?
serious?
Yeah, dark matter.
We don't know
That's
enough
crazy.
about
He did it with cancer. Cancer, George Blumenthal and Joel Primack, they were both physics faculty, and they did the research. They published the research on dark matter.
So Bloomy's not so bad.
Yeah, I guess not.
He's more like a figurehead, isn't he?
Yeah.
Yeah, he's good.
Alright, so is there any other important ones? Not really. Yeah, the Golgi apparatus is what's responsible for kind of modifying proteins, you know?
That's the wiggly one.
It's like, you know, I'm trying to relate it to. It's like, you know, proteins don't come out like perfect, you know what I mean? Like they need like a little haircut, you know what I mean? It's essentially what the Golgi is doing, you know? Like the proteins are like kind of ugly, you know what I mean? They come out, there's some other crap on there. There's probably like some sugars that are attached to it. There's probably some like, you know, some beta linkages that are on there and they just need to be trimmed off, and that's what the Golgi does. Alright. What is this? I don't know what this is.
That's his homework.
What's that? This is the
Craig
homework?
Venters.
Oh, this is the Craig Venters homework. Oh, cool. I get to tell you guys about your homework. All right. So,
What is this?
so you guys are going to watch another cool video about a guy named Craig Venter, who was this crazy dude, who essentially, what he did was he said, you know, what is to stop a certain cell, like a bacterial cell or any living organism, why can't we modify artificially? Why can't we modify that cell? So normally, when there's different types of genetic modification that's going on this campus, in the bioengineering department, or computer science, or whatever, I meant to say bioinformatics, what they're doing is they're taking DNA from another organism, and they're actually inserting that DNA into some sort of cell, and that cell is changing. So an example of this is like, well, how do we visualize cells? We get this protein, we get this gene from a jellyfish called green fluorescent protein. Has anyone ever heard of the green fluorescent protein? Yeah, some of you have. Okay, so that's what makes this jellyfish essentially fluorescent green. So it's like, well, how can we visualize these proteins? How can we visualize different processes going on in cells? Well, let's get this gene from a jellyfish called green fluorescent protein. Let's get that DNA and let's cut the DNA of the organism we're trying to study and insert that gene right into there. And then all of a sudden, all this green fluorescent protein is being expressed, and we can see it all over the cell. Bam. What's not so special about that is that we've taken DNA from a different organism, and we've put it into organism A, we've taken that DNA, put it into organism B. Now, what Craig Venter did was he had a bacteria, and they worked for like 15 years to synthesize their own DNA, which means that they did not use it from another organism. They literally went to the chemistry lab and artificially made a genome themselves. He then got that bacteria, inserted his artificial genome into that bacteria, and then watched that bacteria change before his eyes. And that's what you guys are going to be looking at. Now, there's huge implications to that I'm not going to go over right now. But the kind of big point is, oh my God, this guy just was able to change life. he was able to reprogram life. Yeah.
Wait, so he
What's your name
actually
again?
Zach.
Zach.
So he actually retroactively like changed the cell and it developed. From
So as
fully
he
developed?
as he got his pipette, and he didn't actually do this, but you know, as he squirted in the DNA into his cell, he watched that cell turn from green to red or whatever it did. You know what I mean? It literally changed
So
before.
extrapolating from that, if you were somehow able to do a more advanced version of what he did, would you just be able to like inject someone else and be like turn them into like I don't know some different genes?
You know, David, I think you would look way better as a female. You know, like I think personally, like I think I'm just gonna go over here and just squirt this into you and change you into a girl. So what are the implications of that? You know, like that's crazy. So that's gonna be your homework. You guys are gonna watch like a 60-minute movie. You guys are gonna have a whole bunch of time to think about it, and then we're gonna have another discussion on it.
On Monday.
Yeah.
So was what he did sort of like similar to GMO crops?
So that's a really good question. The difference between GMO, who knows kind of the difference between GMO? Yeah, exactly.
So, or I'm talking about the GMOs. I mean, it's done beforehand. You just take the seed, you modify the DNA, and then you plant it. And they've actually made like Terminator seeds, so you will have to buy it from Monsanto or whatever. But basically, it's done beforehand. But that seems like it's like doing it actively. It'd be the same as like going up to a tree and being like, okay, it's an apple tree. Now I'm gonna make an orange tree.
So that's good. The main difference, I think you're onto it. The main difference is that GMO is still taking the DNA from another organism. And like we've never been able to just like make our own DNA exactly customizable. And I guess I'll let you guys in another cool part of this. So he synthesized his own genome, right? And there are different parts of our DNA that don't code for anything. They are called non-coding regions or introns. And he was able to actually write paragraphs of like philosophical logic into his genome of like the implications of what he had just done. He literally coded into his own genome all sorts of like stuff. So the reason why he did that, it was called a watermark. The reason why he did that was so if that bacteria ever replicated and went out, someone could trace it and be like, oh my god, that's Craig Venture's organism that he just made. So
And another difference is like, so Craig Ventura synthesized it from scratch, but then the GMOs are taking something that's already existing and just changing one part of that.
exactly.
So, and then, you know, they can like patent DNA somehow, even though evolution naturally, or, you know, DNA evolves. So,
What happened to like this?
yeah.
So if you guys didn't understand that, that's okay, because you guys are going to watch it on your homework, and Emil's laughing at me. So you can just keep laughing. All right. How was DNA discovered? This is a cool story. We won't go over it in too much detail, but there's a guy named Watson and Crick, and they discovered it back in the day. They, with the help of someone who didn't get a lot of credit, who was Rosalind Franklin. Watson and Crick won the Nobel Prize for discovering DNA, and they never could have done it without this photograph that was done by a woman named Rosalind Franklin. And she never got credit because she later died from all the radiation. She developed cancer and later died because back in those days they didn't know about radiation and taking X-rays. But there's a really good movie called Double Helix about how Watson and Crick discovered DNA, and we should watch it sometime.
I want to make a little thing.
Yeah,
So like,
make a comment.
so this is like a 2D image. So it's like they have this three-dimensional DNA structure, and then they shine light. It's called like diffraction, and then they get this diffraction pattern. And from this 2D diffraction pattern, they can extrapolate, you know, what the 3D shape was. So that's pretty cool. And then they can do the same thing with proteins. Like, how do I know like what the protein three-dimensional structure is? The way you do that is you make protein crystals. So you like freeze the protein, and then they form these periodic crystals, so they're all shaped in the same way. And then once it's a periodic array, you can shine light and like get like a 2D image like that, and then you can extrapolate the three-dimensional structure of the protein. So that's like a biophysics tools. Like this is like really related to biophysics, optics, and like understanding like the three-dimensional structures from diffraction patterns.
Zach.
Is it true that the double helix structure was first visualized by Watson and Crick when they were on LSD?
So, So I've heard that rumor before. Whenever I hear like rumors like that, one of the rumors is like DMT is released when you die. Not true. But that rumor is unconfirmed. It might be true. So there's a guy named David Presti who's going to be a presenter at our conference, and he is my main go-to man when we have one of these, because he usually knows everyone. He's like, oh, I've met Crick, and I've asked him. But actually, people have asked Crick whether his being on LSD helped him discover DNA. And there was like mixed results. At one point he said like, in the beginning, he was like, yeah, not really. And then towards the end he was like, yeah, like it did. So no one really knows, I don't think he was like, it becomes very like he wasn't really on LSD when he physically discovered it, but he might have like been on it at some point before and then kind of got inspired. But I will tell you guys one thing, if you guys are eager to hear about LSD and scientific discoveries. There was a discovery of the most widely used biology technique ever, and it's called polymerase chain reaction. Who's heard of that? PCR. Can someone explain what PCR is used for? I don't really know.
You find a sequence of DNA that you're interested in, and you just make it really, really, you make lots of it. So that way you can look at it really easily, basically.
So it's a way of replicating DNA really fast. So we can get, say we have one gene that we really like and we want to make lots of copies of it, we use it. I can guarantee you right now there's polymerase chain reaction running in the biology on Science Hill right now. The guy who discovered it, I don't remember his name, totally crazy dude, claims 100% that it was LSD. Fun fact. Okay. Yeah, so this is what we were talking about. How that photo was extremely important.